Tonight's lecture is over Chapter 17, and Chapter 17 covers the special senses. So this chapter is really an extension of the nervous system. Recall from the nervous system that sensory neurons help us to perceive the world around us, and so the special senses help us to sense the world around us. Sensory system. Our body has a variety of ways that it uses to perceive the world around us. There are many senses that constantly gather information about our environment, but the best 5 known senses are the senses of touch, taste, hearing, sight, and smell. Each sense organ is composed of special cells that are responsible for gathering specific data. These sense organs are connected to our brain by the peripheral nervous system, allowing us to process external information. Among our sensory organs, the organs responsible for sight and hearing are considered to be the most well-developed. So there are 2 types of sense. A general sense, where we can sense pain, touch, pressure, proprioception, the sense of position and orientation, and these are widely distributed throughout the body. And then on the other hand, we have the special senses, which include taste, smell, hearing, and sight. And these receptors are localized in the head. Furthermore, we have types of sense receptors. We have the mechanoreceptors, which include touch and hearing, thermoreceptors, where we sense heat, chemoreceptors, which we taste and smell with, photoreceptors, which help us to sense light, nociceptors, which help us to sense pain and damage, and then the sense of balance and equilibrium, which is found in the ear. So the first special sense that we'll discuss tonight is the sense of smell, or olfaction. And this part of the lecture begins on page 601 of your textbook. But smell and taste are chemical senses. Furthermore, the human nose contains 10 million to 100 million receptors for smell in the olfactory epithelium of the superior part of the nasal cavity. The olfactory epithelium covers the inferior surface of the cribriform plate of the ethmoid bone of the skull and extends along the superior nasal concha. So this next slide is figure 17.1 on page 601. And this shows the olfactory epithelium consists of olfactory sensory neurons, supporting epithelial cells, and basal epithelial cells. So this is just a sagittal view of that, and we can see there in the little box that the nerve, the olfactory 1 nerve, is sitting on top of the ethmoid bone, and then that purple area there is going to be heavily saturated with the olfactory epithelium. So there's really 3 different types of cells that are responsible for olfaction. And we see that the first one is the olfactory receptor cells, then we have supporting cells, and then we have basal cells. So the olfactory receptor cells, those are responsible for your sense of smell. So when the chemical or the smell comes in contact with those cells, that's going to send the signal to the brain telling you what you are smelling. The supporting cells, on the other hand, they're going to provide nourishment and physical support. And then the basal cells are basically like stem cells, so when other cells die, the basal cells will help to replenish dead and dying cells. And this slide is just a continuation of the previous slide. It's just that box has been magnified, and we can see the olfactory bulb, which is part of the neuron that's sitting on top of the cribriform plate of the ethmoid bone. And in that cribriform plate, we see we've got the foramina where the axons are going to pass through. And we can see that there are cells in there in the olfactory epithelium that are going to sense the smell and relay that information back to the neuron to take it to the brain. This slide also includes a light micrograph image of this part of the body. So you can see that in microscopic detail. And this is on page 602, figure 17.1 continued. So olfaction, or the sense of smell. Well, we talked about the supporting cells a little bit, and they're just columnar epithelium. They're located in the mucous membrane lining the nose. They're used for physical support, nourishment, and electrical insulation for the olfactory receptor cells. And remember, we also talked about the basal stem cells, and these cells are going to undergo mitosis to replace the olfactory receptor cells. And then we've also got these things called Bowman's glands or olfactory glands. And these are the glands that produce mucus that is used to dissolve the odor molecules so that transduction may occur. Now, transduction is what happens, what we call the process of turning a chemical smell, for instance, into an electrical impulse action potential, for instance. So when I smell fresh baked cookies, and that is a chemical signal that's entering my nose, coming into contact with my olfactory receptors, and then transduction is occurring because those receptors are converting that into an action potential or an electrical impulse to send through the nerve to the brain. So continuing on with the olfaction, or the sense of smell, when you smell something, receptors in the nasal mucosa send impulses along the branches of the olfactory one nerve. And these will go through the cribriform plate. We talked about that a couple slides ago. And once it crosses through the cribriform plate, it's going to synapse at the olfactory bulb. And then those impulses will travel along an olfactory tract, which is going to send this information to your brain. And then the interpretation in the primary olfactory area in the cerebral cortex, which is in the temporal lobe, occurs. So we're still on page 602, and we're still discussing figure 17.1, but this figure kind of puts the olfaction sensory pathway together for you. So there on the bottom left, we've already seen this image, but you can see that the smell is going to enter the nose, and then that's going to stimulate the olfactory epithelium, and that's going to send impulses through the axons that pass through the cribriform plate and synapse with the olfactory bulb. And then we can see the olfactory tract there, and that is going to take the signal into the temporal lobe of the cerebral cortex. And that is the primary olfaction area of the brain. So on this next slide, we talk about the olfactory transduction. And remember, olfactory transduction is the act of your body taking a chemical and converting that into an electrical impulse. And olfactory means that it's specific to smell. So when there is a smell, we call that an odorant. So when the odorant molecule binds to the olfactory receptor, which is a protein, then that's going to trigger this action of transduction where the chemical is converted into an electrical impulse. And this involves a reaction with something that's called cyclic AMP, and this is what causes the depolarization. And once the depolarization occurs, we have an action potential, and that action potential is going to travel to the primary olfactory area of the temporal lobe. But there's one more step to this process, and the impulse must, from the temporal lobe, it must travel to the frontal lobe, or the orbitofrontal area of the frontal lobe for the odor identification. So what happens is when the signal gets to the temporal lobe, there's not you can't identify the signal you can't tell what the smell is. That's in order to identify the smell, it has to be sent on to this frontal lobe. So once it gets to the frontal lobe, then you can say, yes, this is a good smell, or this is a bad smell, or this smells like fresh baked cookies, or this smells like seafood. So that frontal lobe is really going to help you to identify what the actual smell is. So this slide is figure 17.2 on page 603. And this is just a cartoon of what we were just describing about how the odorant molecule, you can see there on the top, the second one from the left, binds to the olfactory receptor protein. And that triggers a sequence of events where cyclical AMP is going to come into play and help open up these sodium ion channels, and that's going to initiate the depolarization of the olfactory receptor cell membrane that's going to trigger the action potential. So on this next slide, we're going to move on to the sense of taste, or gustation. So like smell, taste is a chemical sense. However, it's simpler than olfaction. There are only 5 primary tastes: sour, sweet, bitter, salty, and umami. Flavors other than umami are combinations of the other 4 primary tastes. Taste buds contain receptors for the sensation of taste. Approximately 10,000 taste buds are found on the tongue, and taste buds are also found on the soft palate, the pharynx, and the epiglottis. And like the sense of olfaction, taste buds contain 3 kinds of epithelial cells: supporting cells, gustatory receptor cells, and basal stem cells. And these types of cells are analogous to the types of cells of the olfactory sense. So this next slide is on page 606, figure 17.3 in your textbook. And in the upper left-hand corner, that's really just the surface of your tongue, or a cartoon of that. So we can see where the taste buds are located in the tongue. And then to the bottom right there, we can see in a magnified view, cartoon view of the structure of a taste bud. So we see the gustatory receptor cell and the basal cell and the supporting cell. And then we have the sensory neurons, and those sensory neurons will take the chemical signal out to wherever it needs to go. So taste buds are located in the elevations on the tongue called the papillae. And there's 3 types of papillae that contain taste buds: the volate papillae, the fungiform papillae, and the foliate papillae. On the other hand, you also have a filiform papillae, and these cover the entire surface of the tongue. Now, what's unique about these is that they do not contain taste buds. These are really just tactile receptors, and what they do is they serve to increase friction and to make it easier for the tongue to move the food within the mouth. So this is also on figure 17.3 on page 606, but here we just see the surface of the tongue and how the various papillae are oriented around the tongue. So I guess the volate papillae, they're going to be the easiest to identify because they have that distinctive V shape, and they're towards the back of the tongue, and they're kind of big compared to some of the other papillae on the tongue. So on this next slide, we talk about the 3 cranial nerves that are involved in the sense of taste. And we can see the anterior 2/3 of the tongue. The nerve responsible for that, for carrying those signals to the brain, are the facial nerve, or the cranial nerve 7. And then the posterior third of the tongue, glossopharyngeal nerve carries the taste information. And then in the epiglottis and the throat, the vagus nerve is going to be responsible for carrying that information. So this cartoon is on page 607, but it's a continuation of figure 17.3. And here we just see the pathway. So when the taste comes into the mouth, depending on what part of what taste bud is being stimulated, there's going to be a signal that's sent through the medulla oblongata up through the thalamus, and then that's going to enter the primary gustatory area of the cerebral cortex. So now we're going to switch gears and start talking about vision. And this is figure 17.4 on page 608. And we can see that this is what we call the electromagnetic spectrum. So we have this thing kind of there in the center that we call visible light, and that's magnified to where we can break visible light down into colors. So 400 nanometers, we're going to see the color violet, and then at 700 nanometers, we're seeing the color red. Now on the left side, or the violet side, the smaller the wavelength, and we'll talk about wavelengths in a second here, but the smaller the wavelength, the more energy that wave is going to have. So the color violet has more energy than the color red. So we humans, we live in this spectrum of visible light, and I should say we see in this spectrum of visible light. So we can't see these other kinds of lights like radio waves, microwaves, infrared rays, UV, X-rays, and gamma rays. So radio waves have less energy than gamma rays. But this is what we call the electromagnetic spectrum, and the light that we can see ranges from about 400 to 700 nanometers in wavelength. So in this slide, we're looking at wavelengths, and this is just defined as the distance between two consecutive peaks of an electromagnetic wave. So this slide is figure 17.5 on page 609. And we could see when we talk about vision, we need to talk about the anatomy of the eye. And the eye is going to have these accessory structures. And these include the eyelids, the eyelashes, the eyebrows, the lacrimal apparatus, and the extrinsic eye muscles. So the palpebral muscles control eye movement and extrinsic eye muscles, and they are responsible for moving the eyeball itself in all directions. The conjunctiva is a thin protective mucous membrane that lines the eyelids and covers the sclera. So if this conjunctiva becomes infected or inflamed, some blood vessels are going to turn the eye into a pinkish color, and then we get something called pink eye or conjunctivitis. And then we have the tarsal plate, which is a fold of connective tissue that gives form to the eyelids. And this contains a row of sebaceous glands that keeps the eyelids from sticking together. So your eyelids make a little bit of oil to prevent them from sticking together. So this cartoon is on page 610, figure 17.6. And this is just a sagittal section of the eye and its accessory structures. So first and foremost, we see the eyelashes that are anterior to the eye. And then we see the little green tarsal glands in there that are going to produce some sebum to help your eyelids from sticking together. And then we see the palpebrae muscles there on the superior side of the eye. And then we have some rectus muscles, both superior and inferior to the eye. So the lacrimal apparatus produces and drains tears, and there is a pathway for tears. First, we have the lacrimal glands, and the lacrimal gland is superior and lateral to the eye. And then we have the lacrimal ducts and the lacrimal puncta, which is the little hole right there in the corner of your eye, and the lacrimal caniculi, the lacrimal sac, and then the nasal lacrimal ducts that carry tears into the nasal cavity. So when you cry, that's why you get a stuffy nose, is because some of the tears are also released into your nasal cavity. So this is still figure 17.6, and we're still on page 610, but we can see our lacrimal gland, which is superior and lateral to our eye. And then we can see the flow of tears over there in the right-hand column. So be familiar with that. So there are 6 extrinsic muscles that move the eyes in almost any direction. And remember, language is important here. So oftentimes you can tell by the name of the muscle what kind of a movement it's going to make. These muscles include the superior rectus, the inferior rectus, the lateral rectus. So think about it. The lateral rectus, they help you to look to the side. The medial rectus is going to allow you to look at your nose. And then the superior oblique. Now this is a little bit different because the superior oblique is going to allow you to look down at your feet. And the inferior oblique is going to allow you to look up at the sky. And then furthermore, the eyeball contains 2 tunics. And a tunic is just a fancy word for a coat. So first there's the fibrous tunic, which includes the cornea and the sclera. The sclera is just the white part of the eye. And then the vascular tunic, which is the choroid, the ciliary body, and the iris. So once again, on this slide, we're back on figure 17.6, still on page 610. And we already looked at this cartoon one time, but this is just kind of a review here where you can see those intrinsic eye muscles and the various parts of the eye that we were talking about on the previous slide. So this cartoon is on page 612, and this is figure 17.7. But here we just see a little bit more detailed version of the previous slide, a little bit different angle too, I think. But look, you can see the sclera, you can see the choroid or the vascular tunic and how it's riddled with blood vessels. And then you can see that retina, which is going to send the signal to the optic nerve. So just go through this slide and become familiar with the various parts that it's labeling for you, so you know the anatomy of the eyeball a little bit better. So in this next slide, we see figure 17.8 on page 613. And the iris is the colored portion of the eyeball, and this controls the size of the pupil. That's based on autonomic reflexes. So you can see the 3 different variations of light. You've got bright light, normal light, and dim light. And in bright light, the pupil is going to constrict. And then in dim light, the pupil is going to dilate. So what the iris is doing, or the circular muscles of the iris are doing, it's helping you to regulate the amount of light that's entering your eye so your eye doesn't get damaged. So the retina lines the posterior 3/4 of the inner layer of the eyeball, and the retina may be viewed using an ophthalmoscope. And the optic nerve, too, is also visible. The point at which the optic nerve exits the eye is the optic disc, and this is a blind spot. So the reason this is a blind spot is because there's no retina covering this part of the eye, so it can't collect light to see. So you do have a blind spot within your eye. And then the exact center of the retina is called the macula lutea. And in the center of the macula lutea, we have the fovea centralis, which is the area of the highest visual acuity. So this next slide is figure 17.9, also on page 613. But the optic disc is the site where the optic nerve exits the eyeball. The fovea centralis is the area of highest visual acuity, which we said on the previous slide. So you can see the fovea centralis is that little dark spot there, and that's in the center of the macula lutea. And then we see over there on the more the left-hand side of the figure that there's that optic disc, which is your blind spot, and then you can see how the retinal blood vessels are going to enter in around the optic disc. So the retina contains sensors or photoreceptors known as rods and cones. Rods help you to see dim light, and cones help to produce color vision. From these sensors, information flows through the outer synaptic layer to the bipolar cells, through the inner synaptic layer to the ganglion cells. The axons of these exit as the optic 2 nerve. So this cartoon is figure 17.10 on page 614. And here it just kind of shows us the structure of the retina. And we can see the cone, which is cone-shaped, and then the rods, which are rod-shaped. You've got your pigmented layer and your outer segments. And that signal is going to be sent down through the horizontal cell and the bipolar cell. It's going to reach the ganglion cell, where it ultimately synapses with the optic 2 nerve. And this is just 17.10 continued, and this is just showing how the optic nerve exits through the optic disc. The eye is divided into an anterior chamber and posterior chamber by the iris. Remember, the iris is just the colored portion of the eyeball. The anterior chamber between the iris and the cornea is filled with what we call the aqueous humor, and that's just a clear, watery substance. The posterior chamber lies behind the iris and in front of the lens. The posterior cavity, the vitreous chamber, is filled with a transparent gelatinous substance that we call the vitreous humor. So in order to see, light passes through the cornea, the anterior chamber, the pupil, the posterior chamber, the lens, the vitreous humor, and then is projected onto the retina. So this cartoon is figure 17.1 on page 616. And this is just a close-up of the eye where we can see the lens and the cornea and how they relate to one another. So this is table 17.1 on figure 617, and this is just a summary of the structure of the eyeball. I should say the structures, plural, of the eyeball. So please go through this table and become familiar with all the aspects of it. So this is figure 17.12 on page 617. Light refracts or bends when it passes through a transparent substance with one density into a second transparent substance with a different density. For instance, a light passing through air and coming into contact with your aqueous humor is going to bend or refract that light. So this bending occurs at the junction of two substances. So images focused on the retina are inverted and right to left reversed due to refraction, and then your brain will correct this image. The lens must accommodate to properly focus the object, and then the image is projected onto the central fovea, which is the site where the vision is the sharpest. So this is also figure 17.12, again on page 617, but we can see that the evergreen tree, when we're looking at that, it's by the time it hits our retina, it's upside down, and then the brain is going to have to correct that image. So normal vision is called emmetropic vision, and this is where the eye will refract light correctly and focus a clear image on the retina. So in cases of myopia or nearsightedness, the eyeball is longer than it should be, and the image converges, narrows down to a sharp focal point in front of the retina. So in other words, the image is still in your eye, it's not at its focal point on the retina, it's at its focal point in front of the retina. So nearsightedness just means that people, they need something to be closer to their eye in order to see it clearly. And these people, I just said that, people who have myopia see close objects sharply but perceive distant objects as blurry. So in order to correct this, we can get glasses, and the kind of glasses that we would get would have to have some sort of a concave lens, and that will correct this type of vision. So this is part of figure 17.13 on page 619. And so you kind of see what's going on here with myopic vision. See letter B, you have the image coming in and reaching a focal point before it's touching the retina. And then when we put the concave lens in front of the eye, we can see that we change the focal point, so the focal point is actually coming in contact with the retina, and that's going to correct the nearsightedness. So in the case of hyperopia, or what we call farsightedness, and this is also known as hypermetropia, the eyeball is shorter than it should be, and the image converges behind the retina. In other words, the image is hitting the retina, but the focal point is actually behind the retina. So these individuals can see distant objects clearly, but they have a difficult time seeing objects that are close up. And to correct this type of vision, we use what we call a convex lens. And here we see, again, figure 17.13 on page 619. And this is just an example of what happens with farsightedness. So you can see on letter D that the focal point actually occurs behind or posterior to the retina. And then the convex lens is going to correct that and help to bring the focal point onto the retina where it needs to be for correct vision. So astigmatism is a condition where either the cornea or the lens, or both, has an irregular curve. So this can cause blurred or distorted vision. And again, we can use corrective lenses to fix this problem. So this cartoon is figure 17.14 on page 620. Rods and cones, the photoreceptors in the retina that convert light energy into neural impulses, were named for their appearance of their outer segments. So if we look at the cartoon, we can see how the cone looks like a cone and how a rod looks like a rod, and we already had mentioned this several slides ago. So rods and cones contain photopigments necessary for the absorption of light that will initiate the events that lead to the production of a receptor potential. So similar to how the chemical signal in tasting and smelling is converted into an electrical signal, the eye takes light energy and converts that into an electrical signal. And then there are pigments in the rods and cones that help it to do that. Rods contain only a pigment called rhodopsin. So rods, they help you to see in dim light. It's more of a black and white thing. You don't really see color with your rods, but like if you're out in the moonlight, and things look they're either black or they're white or they're any kind of shade of gray in between black and white. So that's why, if you ever noticed, if you're out and it's dark and maybe there's a quarter of a moon out or a half of a moon, you can't always see colors clearly, and that's because your rods. Now, your cones, on the other hand, have 3 different photopigments in them, and one for each of the 3 types of cones: red, green, and blue. So the cones really help you to see color, or vivid color. And then the photopigments respond to light in a cyclical process, which we'll talk about on the next slide. So this cartoon is at figure 17.15 on page 621. But we can see when the light comes in that there's isomerization of the retinal. And then that leads to the transretinal that separates from the opsin. And this is a process that we call bleaching. And then we have what we call a colorless opsin. Oh, and one other thing I wanted to point out is that the blue arrows indicate the bleaching steps, and then the black arrows, as I said, after the colorless opsin, step 3 there, where the retinal isomerase converts to the trans to cis retinal. The black arrows represent the regeneration steps. So remember that the process of photopigments is cyclical. So light adaptation occurs when an individual moves from dark surroundings to light ones, and this can occur in seconds. Dark adaptation takes place when one moves from a lightened area to a dark one. This takes minutes to complete. Part of this difference is related to the rates of bleaching and regeneration of photopigments in rods and cones. Light causes rod photoreceptors to decrease their release of inhibitory neurotransmitter glutamate. So the neural pathway for vision begins with the rods and cones that convert light energy into neural signals that are directed into the optic 2 nerves. And this is figure 17.17 on page 623, but here we see the visual pathway where we start with the optic 2 nerve, we go into the optic chiasm, the optic tract, the lateral geniculate nucleus of the thalamus, the optic radiation, and then the primary visual area of the cerebral cortex and the occipital lobe of the brain. So continuing with page 623, figure 17.17, we see that the anterior location of our eyes leads to visual field overlap, and this gives us what we call binocular vision. So the 2 visual fields of each eye are nasal and temporal. The visual information from the right half of each visual field travels to the left side of the brain, and the visual information from the left half of each visual field travels to the right side of the brain. So here's a continuation of figure 17.17, and this is on page 624. But we just see that the left eye and its pathway and the right eye and its pathway, and the optic nerve 2, it will actually, from each eye, will join together at the optic chiasm, and then once again they split, and then you can see that the right eye signal is going to the left side of the brain, and the left eye signal is going to the right side of the brain. So this brings us to hearing and equilibrium. The ear is responsible for both of these special senses. The transduction of sound vibrations by the ear's sensory receptors into electrical signals is 1,000 times faster than the response to light by the eye's photoreceptors. Well, you've heard the old adage, the hand is quicker than the eye. Well, I guess the ear is quicker than the eye, too. The ear also contains receptors for equilibrium, and all I mean by this is balance. So ear actually helps you to balance. And then the ear is divided into 3 regions. That's the external ear, the middle ear, and the internal ear. So this cartoon is figure 17.18 on page 626. But we can see there in the lower left that when we talk about the three parts of the ear, the external ear is in the gold, and then the middle ear is in the blue, and the internal ear is in the pinkish color. And then we look to the right there at the blown up image of the ear, and we could see its various anatomical features in greater detail. So the external or outer ear contains what we call the auricle or the pinna. So part of the auricle would be your earlobe, and the earlobe is the part of your ear that you like to get pierced, or it's popular to get pierced, and that's all part of your auricle. And then that leads to the external auditory canal, and then that will lead to the tympanic membrane or the eardrum. So if you were to put the Q-tip in your ear to clean your ear, and eventually you get to where you can't move the Q-tip into the ear anymore, that means you're butting up against your tympanic membrane. Now the auricle is going to capture the sound, and it's going to funnel it into the external auditory canal, and the external auditory canal transmits sound into the eardrum. Also, the ceruminous glands, if you remember, we talked about these in 221. I believe it was either chapter 1 or chapter 4. But the ceruminous glands secrete cerumen, and that's just a fancy word for earwax, and this is just designed to protect the canal and the eardrum. And also the cerumen helps to repel insects. So in the middle ear, we have three auditory ossicles, and these are the smallest and most dense bones in the body. They are the malleus, the incus, and the stapes. So sound vibrations are transmitted from the eardrum through these three bones to what we call the oval window into which the stapes fits. The auditory tube, or the Eustachian tube, extends from the middle ear into the nasal pharynx, and this helps to regulate air pressure in the middle ear. So if you've ever been on a plane, or if you've ever went up the elevator in the Sears Tower, and when your ears start to pop, and sometimes you have to open your mouth and kind of move your jaw a little bit funny, or some people will chew gum to offset that. Well, that's just an example of how the auditory tube is helping to regulate pressure in your middle ear. So this is figure 17.19 on page 627. And if we look in the middle ear, we see the kind of darker yellow figures where they're labeled malleus, incus, and stapes. So those are the bones of the ear. And then we can see there in the lower right corner of this cartoon the auditory tube. So the inner ear, or the labyrinth, contains the cochlea, which translates vibrations into neural impulses that the brain can interpret as sound. And then it also has something called semicircular canals and a vestibule, which is not listed on the slide. And those work with the cerebellum for balance and equilibrium. So this cartoon is figure 17.21 on page 629. Here is just a cartoon of the inner ear, and we can see there in the dark orange, we have the dark orange surrounded by the kind of the lighter orange, we have the cochlea, and then notice where the vestibule is, and then we've got the semicircular canals, which are kind of protruding there above or superior to the vestibule. So vibrations are transmitted from the stapes through the oval window, whose vibrations are about 20 times more vigorous than those of the tympanic membrane, to the cochlea as fluid pressure waves are transmitted into the perilymph of the scala vestibuli. From here, pressure waves travel to the scala tympani and then to the round window which bulges into the middle ear. So this cartoon is on page 629, figure 17.21. And here we just see the up-close cartoon of the inner ear, where we can see the cochlea and then the scala vestibuli and the pathway that notice the arrows that the sound will travel through the cochlea. So this cartoon is on page 630, figure 17.21, and we can see that the inside of the cochlea, or one turn of the cochlea, and where the sound will be converted into an electrical impulse there through the cochlear branch of the vestibular cochlear 8 nerve. So pressure waves travel from the scala vestibuli to the vestibular membrane to the endolymph of the cochlear duct. And then the basilar membrane vibrates. This moves the hair cells of the spiral organ, or the organ of Corti, against the tectorial membrane. And these cells generate nerve impulses in the cochlear nerve fibers. So again, this is still on page 630, figure 17.21. But here we see the tectorial membrane, and then the outer hair cells and the supporting cells, the inner hair cells, and this basilar membrane. And then we can see the sensory and motor fibers in the cochlear branch of the vestibular cochlear nerve. So that's where, as I said in the previous slide, that's where the sound is being converted into the electrical energy to create a nerve impulse so your brain can interpret the sound. So this cartoon is figure 17.22 on page 631, and this just gives us kind of the broad overview of the hearing process, where through the external auditory canal, the sound is going to reach the tympanic membrane, and that's going to interact with the malleus, the incus, and the stapes, which is going to vibrate into the oval window, and that sound is going to move through the cochlea and the organ of Corti until it synapses with the vestibular cochlear nerve and creates an electrical impulse that your brain can interpret as sound. So the cochlear nerve fibers form the cochlear branch of the vestibulocochlear nerve. The axons synapse with neurons in the cochlear nuclei in the medulla oblongata. The impulses travel to the medial geniculate nucleus of the thalamus and end in the primary auditory area of the cerebral cortex in the temporal lobe. And here we see a cartoon of that pathway. This is figure 17.24 on page 634. But from the hair cells of the cochlea, the auditory information is conveyed along the cochlear branch of the vestibular cochlear nerve and then to the brainstem, thalamus, and cerebral cortex. So when we talk about equilibrium, there are two forms of equilibrium. One is static equilibrium, and this is the maintenance of the body's position relative to the force of gravity. And then we also have dynamic equilibrium, and this is the maintenance of the body's position in response to sudden movements. So the vestibular apparatus, these are the organs that maintain equilibrium, and we can further break down the vestibular apparatus into the saccule, the utricle, and the semicircular canals. Now, otoliths or otolithic organs, which include the saccule and the utricle, are calcium otoliths are calcium carbonate crystals. So what this means is that you basically have crystals floating around in your ear. The walls of the utricle and the saccule contain a macula, and the two macula are receptors for the static equilibrium. So the otolithic membrane sits on top of the macula, and movement of the head causes gravity to move it down over hair cells. And that's what's going to create the action potential, so you know where you are in space. The hair cell synapse with neurons in the vestibular branch of the vestibulocochlear 8 nerve. So this is figure 17.25 on page 635, and if we look on the left there, we can see the structure of the inner ear, and it has the saccule highlighted in blue, and then that's magnified so you can see that you have these hair bundles that are connected to nerves, and that when those hair bundles are moved, those hairs are moved, then the action potential is going to be generated, and that's going to send the signal to your brain. And this cartoon is also the same figure, 17.25, but we can see the otoliths or the crystals above there, and the hair cells. And so when those crystals become activated, that's going to vibrate or move your hair cells, and that's going to create the action potential. Three semicircular canals are responsible for dynamic equilibrium. The ducts lie at right angles to each other, which allow for rotational acceleration and deceleration, or deceleration, I should say. So, for instance, when you're in a car and you can tell that you're moving, or you can tell that you're speeding up or slowing down, that has to do with your dynamic equilibrium. An ampulla in each canal contains the crista with a group of hair cells, and its movement of the head affects the endolymph and hair cells. So endolymph is a kind of fluid, and this generates a potential leading to nerve impulses that travel along the vestibular branch of the vestibular cochlear nerve. So this cartoon is figure 17.26 on page 636, but we can see the ampulla and then the crista. And if you were to move your head, then that would stimulate, or it would create movement in the hair cells, and that's going to create an action potential in the nerve. And in this cartoon, we're still on figure 17.26, but now we're on page 637. So you can see the cartoon, the person down at the bottom, their head is in a still position, and then look at how the cupula is kind of upright, and then as the head rotates in one direction, the cupula is dragged through the endolymph and bent in the opposite direction, and then that is going to move the hair cells, and that will send the signal to your brain to help you to maintain your dynamic equilibrium. And this cartoon is figure 17.27 on page 638, and this just shows us the pathway from the ear, the sound or the equilibrium, how that is going to move through the brain. And we can see that it starts with the vestibular branch of the vestibulocochlear nerve, and that's going to go to the vestibular nuclei, and then it's going to go to the thalamus, and then that from the thalamus is going to go to the vestibular area in the cerebral cortex. So the next several slides relate to table 17.2 on page 639. And on this first one, you can see that it just summarizes the external outer ear. And if we move to the next slide, we see that we have the inner ear mapped out for us in a very nice summary. And then finally, we have the inner ear, and you can see all of its complexity and different parts. So make sure you review that table. So now we'll go into the development of the eyes and the ears. The eyes begin to develop about 22 days after fertilization. The ectoderm of the forebrain forms the optic grooves, and then they will become the optic vesicles. The optic vesicles reach the surface ectoderm, which thickens to form the lens placodes. The distal portion of the optic vesicles forms the optic cups, and they remain attached to the proencephalon by optic stalks. So this cartoon shows the development of the eyes and ears, and this is figure 17.28 on page 640. But you can see that after about 22 days, the eye starts to develop, and it develops from the ectoderm. And then just follow the cartoon here for more detail about what's happening at each point of the process. As far as the ear goes, the internal ears will develop first, and this also begins about 22 days after fertilization. The surface ectoderm thickens to form the optic placodes that appear on either side of the hindbrain. They form the optic pits that pinch off to form the optic vesicles. And this cartoon is figure 17.29 on page 641. And here we just see how the ear is developing over a period of days. So, aging and the special senses. Smell and taste are not affected by aging until around the age of 50, when the gradual loss of receptors and the slower rate of regeneration start to have an effect. As far as vision goes, the lens begins to lose elasticity and has difficulty focusing on close objects. And around 40, muscles of the iris start to weaken and react more slowly to light and dark, causing elderly people to have difficulty adjusting to changes in lighting. Furthermore, retinal diseases such as macular disease, detached retina, and glaucoma occur more frequently in the elderly. By about age 60, approximately 25% of individuals will experience a noticeable hearing loss. Tinnitus, or ringing in the ears, and vestibular imbalance also occur more frequently in the elderly. And that concludes our lecture for this evening.